Hi. So I'd like to start by thanking the organizers for the invitation, and Chris especially. I'm probably by far and away the most theoretical person in the room, although I would be happy to prove wrong later. I'm in a mathematical sciences department. I was trained as an algebraic topologist, and then through an extensive sequence of coincidences ended up studying neuroscience. So as a result of my mathematical background, I have a tendency for overly complicated titles. And after I submitted this title, I realized that this was unbelievably ambitious for 25 minutes. So I'm going to go ahead and modify my title just a little. Localizing will be made parenthetical throughout. Okay, so I want to talk a little bit on, there was briefly some discussion about algorithms for actually controlling circuits. There's been a lot of really great questions about data and all of this massive data, right? All these very beautiful techniques that have been presented this morning for gathering really, really, really huge amounts of neural data, multimodal data, and you run into the question, which has been asked repeatedly: what do you do with all this data? And hopefully you build models. That's a lot of the people here are modelers. So I want to think about topology in particular as a tool for neuroscience, because I think it's a very powerful tool that not a lot of people are really aware of. It's an emerging tool. I know a lot of people in the neuroscience community say the word topology, and they mean networks, and those are topological. But the topologists would beg to differ. So I want to tell you a little bit about topology and what it's good for, where it came from. So I want to start with this simple dynamic system. So imagine this is a biology group, so I'm going to tell you some physics. Imagine a universe with 2 planets in it. And those planets are orbiting each other in a circle. This is a stable dynamic system. Okay, they'll orbit forever because they only have, if they're appropriate velocity, they will orbit forever. So in this universe, you might imagine that the scientists would rapidly discover that there's exactly one thing to look at, and they would build telescopes that look only at the other planet, right? So this is our setup, and now imagine we introduce a slight perturbation into the system. So I'm going to add another planet. I'm going to put it on the axis around which the 2 planets revolve. All right. So this is a question that Henry Poincaré, who is the person who invented topology, he started the whole game. This is an example he came up with, which I think is extremely interesting. So you put this planet on that axis somewhere, and of course it'll start accelerating downward because there's gravitational pull to the other 2 planets. The 2 planets will continue orbiting because, right, they're only being pulled upward. It'll pass between the 2 planets, and at that moment, the scientists will say, "Oh, there's something else in the universe. That's exciting. What was that?" But then it's gone. Okay. And now they wait a little while. They have some discussions about whether their instruments were wrong, and boom, it's back. Okay, and then it leaves again. And this will continue, right, for a long, long time. Question: how many observations do they need to make of the planet going by before they can tell you when it's going to come back? So there are a lot of modelers in the room. Does anybody have a guess?
Greater than 1.
Greater than 1, definitely greater than 1. So 2. It turns out this is a trick question. It's impossible. You can never find out in this tiny system with 2 planets, with 3 planets in the entire universe. You can never make a precise prediction about when that red planet's coming back. It's because the plane in which the blue planets are orbiting is perturbed by the existence of the 3rd planet, and so they oscillate in an irregular fashion. So you probably have heard that the 3-body problem is not solvable. Even this extremely simple 3-body problem is not solvable. That's a problem if you're going to model.
But approximately.
Yeah. that's why Henry Poincaré is a very smart man, was a very smart man. So even if we know everything, deterministic dynamics, you have as much data as you want. It's impossible to accurately fit a model of dynamics capable of making precise predictions. It can't be done. That's bad news. So this is my sales pitch. Anyway, you can do it approximately. Applied topology is this new, exciting area of mathematics, about 20 years old, although there have been applications of topology going back through the ages. The new version of applied topology really came about 20 years ago. And the idea is that we have a lot of data, and a lot of the time the specific numeric predictions you want to make are just impossible to make. You can't make them. You don't, even if you have everything that you could ask for, your favorite wish list, you can't do it. So what we want to do is we want to create qualitative measures, which is exactly what the people in this room do when they're making experimental decisions or making design decisions. You say, the system behaves like this. I have place cells are my favorite example. They record locations in space, right? So you build a model and it's a little bump a little bump in the environment, and that's where the neuron is active. That's a qualitative model. You don't actually think that there's a Gaussian centered at the point. Nobody thinks that, right? So, but the qualitative features should be approximately correct. And topology is sort of the language for doing this. And what I'm going to do this morning is sort of promise you a lot of things that we haven't built yet. But I will point out a few things that we have built, and also some things that people are building right now that I think would be very useful to this community. So here are some topological tools. These are the three I'm going to spend a bit of time with. So simplicial complexes are our bread and butter. These are ways of recording higher order structure in observed data. And there is a tool called persistent homology, which I could take easily full 25 minutes and probably 6 more hours explaining to you. I will wave my hand and say it exists. I'm going to tell you about some work by a group at Rutgers, led by Konstantin Mashaikov, something he calls the database of dynamics. And this is essentially the idea that because we can't give you accurate, right, your measurements aren't accurate and our models aren't accurate. So why would we expect the state to be accurate? So let's decompose the state space and maybe the parameter space into a bunch of very tiny regions. And then we're going to study transitions between those regions as opposed to trying to study specific states of the system. And it turns out that this gives you indices, which is a mathematical term for some numbers that are associated in some very, very highbrow way to the dynamics, which then characterize how the dynamics in those regions work. And then I'm going to tell you about something I'm working on right now with some folks at the human cognitive level, but I think, it's a shame I don't have any optogenetic data to run it on, because if only there were some people in this room that could give me some, that measure population time series and give you a very robust notions of relationships between the elements in the time series. Before I go on to those, I want to tell you about some things that didn't make my list. I tacked these on last night, so I don't have a ton of, I don't have pictures for you. But I wanted to say that these are tools that I think people might be interested in. So Mapper is very famous. Many of you might have heard of Mapper because it's in all the journals right now. Every couple days I get another email that somebody has used Mapper for something. So this is by the Carlson group at Stanford, who were some of the first applied topologists. And the idea is to take high-dimensional data, and instead of doing something like dimension reduction, which crushes the structure, you instead look for the structure in the high-dimensional data in a way which gives you a summary of fine features. So you should think about this as sort of clustering without losing the shape of the data. And again, I'm sorry, I'm not going to give you a lot of details about these things, but if they sound interesting, I'm more than happy to point you to papers and things. So just grab me offline. Control bundles are a really great idea. I have a bunch, imagine I have network dynamics, right? Many people in here are interested in network dynamics. We can think about, it's really hard to understand how the various pieces of the network are talking to each other. So what if I just assume everything in the network is its own little control system? Right, then we can apply the kind of tools that John was telling us about, and I can study each of those control systems individually, and then I can try to enforce global consistency on the entire thing. This is only being done in theory. No one has ever applied this to anything. It's just a giant tool sitting out there in the universe. All these are giant tools. Cellular sheaves are the general framework for local to global structure inference. We all believe the brain does this. This is the mathematical language for doing this. Again, it's sort of sitting around waiting to be used. And then there's this very nice tool that I think a lot of you might be interested in called unimodal category, which is just a very simple thing. I'll give you a time series, say calcium trace. Ask, what's the minimum number of unimodal events that created this time series? Right? It's a simple question. The number of spikes that I saw or the number of whatever. It turns out that you can compute this. You can compute it as fast as the time series is being produced. And it gives you a very, very simple measure of the complexity of activity. So these are the sorts of tools that topologists are producing. They work at this level, this very conceptual level, right? They're not, let me give you an exact specific dynamic formula for what's going on. Let me give you a qualitative picture of what's happening in your system. I have a couple more really quick. So a couple people this morning at breakfast were talking about the fact that it's sort of a shame that you take dimensionality reduction techniques and they give you back a flat answer, right? So if you have the Swiss roll, you can flatten it out. But if you have a circle, you're gonna try to flatten it out. You're gonna get a line with the ends broken. So it turns out that there's topological methods that are canonical for assigning manifold, or really what we say projective coordinates to data. These are really natural in neuroscience, right? You have edge detection in V1. So that's what mathematicians would call the projective plane, if you've ever heard of that. So there's natural coordinate systems in the brain that sort of fit these things, and topologists have ways of developing these things. And then there is a group working with the Blue Brain Project over in Switzerland who have been using the fact that they can cheat because they have structure on which their dynamics are modeled to do characterizing the relationship between sequences of cell firing and the physical structures. And all these things are essentially just these same qualitative tools that I'm going to tell you about. And I could go for another hour. So I want to talk about simplicial complexes first, because these are so simple that it's a shame that more people aren't using them. So whenever you see a population of things being active, there are all sorts of ways that you might characterize it, right? Coactivity is the really easy one, right? These things are all active at the same time. Or correlations, strong group correlations, that sort of thing. Okay, and I was delighted to learn that we can now observe colorful neural activity, so this picture is no longer theoretical. We can instantiate it. So if I had a green neuron and a cyan neuron and a blue neuron and a red neuron, right, I look at their activity patterns. If you were going to encode this as a graph, you might say, oh, well, I've got coactive pairs, right? And so I would end up with two triangles of coactive pairs, right? Because that third event, I would get 3 pairs of coactive neurons, which is losing information, right? We know that the group coactivity, the population coding, is at least at some level important. So we should encode it as an object, and we can do that. Simplicial complexes are literally just the idea that you should keep that information. It's a very simple idea. For some reason, it doesn't get used a lot. We have tools, we have computational tools that do this, all right? If I then thought maybe these things came from receptive fields, well, look, what have I done? I've encoded the fact that these 3 receptive fields overlap, right? So again, natural qualitative structure that we normally think about in these things. And then if I have one of these things, I can try to embed it, and I can ask, imagine for a moment I were recording from hippocampus because I like place cells. What was the shape of the environment in which the animal was moving? Well, if it were a graph, I wouldn't be able to rule out that there were obstacles in between every single one of those little triangles because I wouldn't know about the triple firing. But here I've got various filled in regions, so I know, there's a hole in there. and I can try to count that hole and let you know what the shape of the environment is. There's some nice work by I hate it when that happens. There's some nice work, nice experimental work about the fact, or it shows that a lot of these, that the firing in the hippocampus under deformation of the environment is in fact invariant. These intersections don't care about the actual shape of the environment, only the relative positions of the cell positions. So in fact, in some sense, this is the right way to think about it. There are intersections or there are not intersections, not these intersections correspond to this location. If it happens that you didn't do this ahead of time, that you just built yourself a network, which a lot of people who do, for example, you might have computed pairwise correlations, we can always just soup it up, take your network that you've built and build a simplicial complex out of it by filling in all the triangles and all the tetrahedra and all the higher dimensional versions of the same thing. Everywhere you can put in a face, you do. So it's just a canonical thing you do. And it turns out that this works out really well. So you can, before I get to the how it works out really well, let me tell you what we count. So we count the holes. Topologists like to count holes. This is our primary tool. And so here I'm going to count those one-dimensional blue cycles that bound those holes. And here we've got a two-dimensional shell bounding a volume. And you can imagine that's, a shell built out of three-dimensional things, enclosing a hypervolume, et cetera, et cetera, et cetera. These are just qualitative features. And right, when I say this has two holes, you can look at it and say, yes, I agree, that has two holes. I don't have to tell you what they are. We might even disagree about what they are, depending on our interpretation of the underlying mathematical structure. So, but we count them and then we record them as this funny little vector of there's one zero-dimensional hole, meaning there's one component, two one-dimensional holes, and a two-dimensional hole. So we can argue about what that means if you like. I don't think it means much. And then you do this funny thing. You say, well, I had a bunch of correlations and I didn't know which ones were particularly significant. And so the normal response is I will just slice it off at some point, make a binary matrix, and off I go. But it turns out you don't have to do that. You could just take all the possible thresholds, right? And then you get a sequence of these objects in sort of in order of increasing relevance. All right. Yeah, so then I can count the holes in all of them. And there are fast algorithms for doing this. And you get these sort of curves that characterize the shape. All right. And you see this is all very nice, hand-wavy stuff. But as Chris said, I make it a point always to apply these things to data to make sure that it's not nonsense. So here is, we took CA1 pyramidal neurons. So this is place cell data, but we really mean pyramidal neurons that aren't pre-selected to be anything. We just have some recordings. And we did this process. We built these complexes. And so this thing on the bottom left here is the data, the curves that you get by counting the holes in the data. Up right are the curves you get by dropping points in space and connecting them based on distance. And you can see they look just about right. In fact, if you overlay these things, they're pretty spot on. And you can integrate them to get a nice simple number so that you can do all of your easy statistics and people will believe you. It turns out that this does work, and it even works when the animal's asleep. So this really is recording this is recording intrinsic structure in the neural activity. All right, it's giving a nice summary. In some understandable sense. We can compare it to models. We can actually discuss the qualitative structure of the encoded information that in the system. Nowhere here did we use the behavioral correlate. All right. Yeah, they're the Betty numbers. I just don't need to add any more terminology to this terrible talk. I do want to add that there is a paper, a very recent paper by Tatiana Sharpie and a student, I believe, that does a similar analysis on olfactory data using essentially this technique and studies sort of a hyperbolic structure there. So essentially you take the qualitative structure of what you believe the information to be, and you can compare it against the encoded information in the system in this way. Okay, so this is the broad strokes. Let me give you a. Sorry, what time do I end? Today is good.
Today's good. I don't know how much later we start.
5 minutes? Okay, thanks. Okay, so let me bump through the database of dynamics here real quick. So the idea here is essentially, like I said, you cut up the state space, and then you study transitions. Okay, and this is work not of mine. This is work of Mashaikov and Spenlove and about a half dozen other folks. All right, but the idea is that you can characterize the flows as a graph, right, between various states. And then there are these topological indices that you can assign to the nodes that tell you, oh, there's a limit cycle here. Oh, there's this sort of thing. Oh, there's that sort of thing. You don't have to know about the exact locations of these things. You don't have to know the exact equations. You can observe the system and extract this sort of thing. If you change the scale, it changes the structure, but it turns out that the fundamental pieces, that there's an attractor and that there are repulsors, that there are paths between them, those things stay constant as you change the scale. So in some sense, these are scale invariant measures. Okay. So I just wanted to, I'm going to go a little bit faster because I've been talking too much. So I want to talk about path signatures, and here's an experiment being done. So this is an actual feedbacky kind of experiment. So this is EEG transcranial magnetic stimulation experiment. So the idea here is that there's a cognitive task being done there, and they are zapping someone's brain while they're also taking EEG signals. So you look at the EEG traces. This is a 32, this is John Medaglia at Drexel University and Martin Wiener at George Mason. So here, on the left, it's pre-stimulation. On the right, it's post-stimulation, right? 32-channel EEG. Everybody see the difference? So if you look really close to the red line where the stimulation happens, you can see a spike in the activity. I'm going to ask, what's the difference between the left and the right? So what's the difference? So I'm just go really faster. So if we change the details of the stimulation, what if, how do I characterize the left and the right there? So various things you could try to do. Event-related potentials is the easy one. You know where the event is happening, right? We know we stimulated it. What happened immediately thereafter? The other ones, Fourier analysis, Granger causality, structural equation modeling, they all require a bunch of things that are definitely not true, right? Gaussian noise is a really big deal. Linearity is a really big deal. Stationarity, we just zapped the system, right? All of these things interfere with the standard analyses you would do with these. So unfortunately, we can't use them. At least we can't trust the conclusions we would draw from them. So let me tell you about a really simple way to do this. So imagine I had two time series like this. You might claim that if the first one was evoked and the second one was observed, that there was a causal relationship there. I would call it a lead-follow relationship because I don't believe in causality in that way. But you're never going to see that in neural data. You might see this. So it turns out that these have the same, if you draw any vertical line, we've just stretched and squished the t-axis there. So we reparameterized the time axis, right? There's still the same lead-follow relationship. That's my claim. When one is going, when the red signal drops, the gold signal drops after it. When the red signal rises, the gold signal rises after it every time. Okay. So what you can do, this is just an observation from like classical mathematics, is you could plot them against each other, and in both cases you get the same picture because you throw away the t-axis. Okay, that area in the middle there, that's called the Levy area. That's a classical thing. When it's big, one of them is following the other. So you can just compute that. It's really easy. This is, by the way, this is what surveyors use to measure the area of a room. They go around the outside. They compute this little integral, and it gives you the area of whatever you've traced around. Okay, and so the advantage here is that if I have a lead follow, I can compute it, and this is, what is this? This is a dot product, right? There's nothing here. So it's really, really fast. It's model free. It's extremely fast. It sees this cyclicity, right? One of them is ahead of the other. But it's not, that was a non-periodic signal, right? But it gives me the same number. So in non-stationary settings, it doesn't matter because I'm model free. So as long as I believe the same things that I believed when I was doing other causality analyses, this is going to say this one is ahead of that one over and over and over. And due to linearity of the integral, we can do a version of PCA to ask, well, what's the strongest causal signal? And here's the best part. As a topologist, the reason I know about this is not because I studied calculus. It's because there's a massive mathematical theory hiding behind it that tells us that this is the simplest version of this. So if I wanted to look at whole brain variations, I don't have to look at individual time series. I could look at time series on the brain, and we can play the same game. So, but of course, that wouldn't fit on these slides. And I didn't know I just needed to talk about it. So what does it see? Well, here are my 3 stimulation points, pre-stimulation, post-stimulation, and I've just measured the pairwise lead coefficient to a bunch of other, and these are the statistically significant ones in a single trial. So we just stimulated the same person over and over and over again. Blue is follow, red is lead. So here we see that in the prefrontal cortex, pre-stimulation, the stimulation node is following a bunch of the other nodes quite strongly, and afterwards it's leading them. So there's an actual change in the way that the electrodes are seeing the signal on the scalp. Similarly, in the other 2, there's substantial changes post-stimulation. So we can actually begin to characterize this structure. This is work in progress, so you would have to argue with my experimental collaborators about what it means. But it is visible in a way that the other methods I named just fail to show you anything. So the idea here is that we perform a calibration by putting you down and doing some stimulations, and then we would choose particular versions we would use our domain knowledge of the situation to choose new stimulation based on what those previous measures told us to do. And then we would use these measures to determine how well that was, how the system structure was changing, or maybe to identify system structure using the tools. So my claim is that we need entirely new quantitative tools to support any sort of real model building in neuroscience. This is my wish list. I think topology covers a lot of it. There are certainly problems with topology. Computationally efficient is not one of our favorite things yet, but we're getting there. But it needs to be interpretable, and that's a real issue for a lot of current quantitative methods. So, all right, and I should say, just to answer one of the questions that I was asked to answer in this session, I think we need to be not necessarily multi-scale at all times, but we need to have tools that are consistent across scales. In other words, that naturally give us the ability to scale up and scale down internal to the tool. And that's one of the things these local to global style tools actually provide us. Okay, so thank you very much. If any of that was interesting to you, we've written a survey intended for people who are not mathematicians but are neuroscientists, which I've given a appropriately serious name. And then there's this bibliography that Chris mentioned. So thank you very much.
Questions? Comments?
So you mentioned that these tools are mainly for qualitative descriptions. Has there been much work done in kind of linking it to quantitative modeling? Because really that's what we need for closed loop control.
Well, so I guess I would claim that, in fact, you don't need that, because when I say qualitative, I don't mean without numbers, I mean without exact numbers, right? So I would say this is larger than that, or this is going in this direction, as opposed to it's converging at exactly log n, right? Because I can't know that, and I can't really know that from data. So there has been comparison to quantitative, and then of course you can compare it to quantitative things simply by dropping your quantitative model into the same machine, right? And saying, is it consistent with the model? One of the things you can say about, one of the ways you can characterize this is that it does a lot of model verification for you by saying it produces the same qualitative structure as the quantitative model that you may have built.
So basically what I'm wondering is if I know there's a hole in my data,
Mm-hmm.
I still need to get the coordinate, I still need to write down. So is that link is what I mean.
I see. Yeah, so that is, so when you build these complexes out of your data, you have the ability to associate any metadata you feel like adding to them. So there is existing software that does a fair amount of extracting specific things, but we're currently in the process of building tools that are more accessible to the general community that will let you say, for example, you know, I started with this, I started with these brain regions, I started with this, you know, whatever it is that this set of neurons, please give me back the pieces that made up this particular feature. Those tools are in development for the general community. You can force it with the current tools, or you can have an expert help you make them work. Yeah, so I should say the potential is there. Many of these tools, like I said, I'm promising many things that aren't yet built. But we'd be happy to have engagement from the community in deciding what needs to be built next and how to, what problems need to be solved, what new tools we should focus on, because we're mathematicians, and as a whole, we're not very good at choosing applicable directions for our work. So.
Is there a generalization of random matrix theory
Yes.
to this?
Yeah, there's a very active community that studies random simplicial complexes. There's been some very, very beautiful work by a gentleman named Matthew Kale at Ohio State University who sort of kicked the whole thing off. There's a group in Israel, Lanil and Meshulam, that do random two-dimensional and three-dimensional simplicial complexes. So there's a lot of random geometric complexes naturally follow random geometric graphs. And a lot of the results that you see from the random graph world generalize very nice. In fact, almost everything that you know about graphs generalizes in many ways to simplicial complexes. So there's a spectral theory, there are random walks, there's dynamics that live naturally on simplicial complexes. So you just take what you know about pairwise things and you imagine that it could work for multiple things, and it does.
That sounds good. I mean, by the way, just as a comment, right? Mm-hmm. I mean, so one often sees papers where you see, a nice two-dimensional plot. There's a big matrix. Guess what? There are some big peaks in it. You diagonalize that. Oh, you say that this is important science associated with these big eigenvalues. Mm-hmm. And I rarely see anybody test that hypothesis against something which was just a random matrix, which generates a few big eigenvalues anytime.
Yeah.
So there's a sort of set of hygiene that we should be applying, I think, to some of the tools that we do to get these things right.
I strongly agree with that comment. There are a lot of basic results in random matrix theory that I think it would be useful for people to be really fully aware of. The existence of the Perron-Frobenius eigenvector, I think, is an important thing that many people should review. It's just there's a very large eigenvector that's always present in random. So that on the top left there, that's Matt Kale's work. If you take a random geometric or random totally IID graph, the Betty numbers look like this.
So my question, I think, is kind of similar. That the topography gives us some wonderful tools to find the lumps in the soup, if you would. But what we need is a complementary mathematics that has hypothesis model constraints in it that can map to that topology, because
yes.
that's what gives the experimentalists then questions, predictive questions
mm-hmm.
that they can go and ask in the biology.
Yes. And that's part of the reason I'm here, because I think the way that you build that, the honest way to build that, right? If I were to randomly go out and start building hypothesis testing methods, they wouldn't be useless. They'd be completely worthless, right? What we need are good, grounded biological questions so that we can determine what hypotheses we would ever test. And once we've got that, then we can start asking questions, right? So doing these random geometric complexes, nobody had really studied them previous to when we wanted to do this paper. So we did a qualitative, we ran the model on it, and now there's a bunch of theory that's following it up and giving us the answers here. But at the time, we made these funny little curves, and that's what people knew, right? So yeah, I think you're very right. We need We've been developing statistics of these things, but we need to know what is needed. So.
Yeah, so in somewhat unrelated topic, there is actually beautiful work by Rohra Badan at Columbia University on RNA-seq data,
Yes.
actually, where they show that random matrices almost fits the whole distribution, but actually the information is where it doesn't fit. I think that work actually could be very, very relevant to these networks here.
Yeah, so Raul does actually uses a lot of these topological methods in his work, right? So yeah, I agree. There's a lot of crossover potential there, and they've done a lot of foundational work trying to sort of sort out how to use these tools on data at the genomic level.